Sathyabama Institute of Science and Technology.
Good morning, everybody. I'm Jesia Persis Preethi, Assistant Professor from the Department of Biotechnology, Sathyabama Institute of Science and Technology, deemed to be university. We'll be learning about DNA replication. DNA is abbreviated as deoxyribonucleic acid. The DNA is a genetic material which is present in the majority of organisms, and DNA stands for deoxyribonucleic acid. And a few viruses, for instance, TMV virus has RNA as a genetic material. Apart from it, majority of the organisms has DNA as the genetic material. And in the evolved eukaryotes and majority of the organisms, RNA acts as a messenger where it plays a role as a catalytic activity and helps in the translation of the protein. And the number of base pairs determines the length of the DNA. And for instance, human DNA in the haploid state tends to have 3.3 to 3.3 into 10 to the power 9 base pairs. This is in the haploid state of the human genome. And bacteriophages like lambda bacteriophage tends to have 48,502 base pairs. And organism like E. coli, microscopic organism like E. coli, tends to have 4.6 into 10 to the power 6 base pairs. DNA replication is a semi-conservative process, and generally we can see the DNA replication happening during mitosis of cell, where a copy of DNA is formed, and during the mitosis, the mitotic spindle fibers, they tend to pull the daughter or a copy of DNA to the either ends, and once they're pulled apart, then the cytokinesis happens, splitting apart, making two daughter cells. The stages of DNA replication. First and foremost, the DNA has a double helical structure was put forth by Watson and Crick, and this double helical structure has to split apart, meaning that the hydrogen bonds need to be broken, therefore it leads to opening of the helix. Now each strand of DNA acts like template for the synthesis of new strand. Replication produce two identical DNA, and each of them has one strand from the parent which has acted like a template, another strand as a daughter strand which has been a complementary strand which is replicated. So diving into the process of replication, the process of replication is a beautifully mediated process in which a lot of enzymes and proteins work together and make sure the copy of DNA is formed. And first and foremost, for the DNA to be replicated, the enzyme that comes into play is primase and helicase. Helicase is the enzyme which tries to break the two strands apart, making a replication fork which will help in the synthesis of daughter strand. And for this daughter strand to be formed, it generally takes place in three phases. The first and foremost phase is known as initiation, elongation, and termination. So in the initiation phase, the primase and helicase comes together, and it helps in creating the replication fork, making the two strands apart. And these two strands, once they're apart, they each of the parent strands act like a template for the daughter strand to be translated. And if we see the initiation process, and first and foremost, if we have to see the initiation process, there are a lot of sites for the replication to take place, which are known as origins, and these sites has a specific coding region which will help to identify the start of the replication, where the proteins immediately come into action and they recruit a lot of enzymes to start this replication fork and creating a replication bubble. And DNA helicase, which I was stating, make sure the strands are broken apart, for which there should be hydrolysis of ATP molecules, and the DNA polymerases immediately come into action, but before which RNA primase adds a primer to the parent strand where the replication of the daughter strand has to take place. and this is where the DNA polymerase comes into action. We can see this in a detailed picture. So for instance, you can see there are two strands which are split apart. One is the leading strand, another one is the lagging strand. The DNA polymerases are designed in such a way that they can read the parent DNA strand from 3' end to 5' end. And therefore, we can see that the daughter strand is being copied from 5' end to 3' end. Each nucleotide is being added from the 5' end to 3' end, and this RNA primase is added in the initial part, and that recruits the DNA polymerases, and DNA polymerases, once they bind, they start adding the nucleotides and start moving towards the end of the fork. And here, once it starts moving from the leading end to the towards the fork, there is always a tension which develops on the strand which is unopened. Therefore, topoisomerases are the enzymes which help in relieving the stress. And now what happens is, as it forms, it can keep on forming, and as the DNA replication fork opens. Whereas in other strand, the 3' end is towards this end and the 5' is towards the other end. So therefore what happens is, as it moves, now the RNA primer is added somewhere in between towards the 3' end, and it keeps on moving. And now what happens is, now the newly formed strand continues to form, and it forms as a discontinuous, as say for instance, now it has formed, let us say. Now newly formed fork is opened from this end. So therefore what happens? It cannot come backwards. Therefore again a new RNA primer is added from this end, and it keeps extending. So it forms like a discontinuous. It forms like discontinuous strands. You can see a discontinuous strand here and discontinuous strand here. So therefore this is called as lagging strand, and the fragments of daughter DNA which is formed are called as Okazaki fragments. And these Okazaki fragments are later on being they are sealed by other polymerases, which we'll see in the future. And if you see this, helicases and single stranded binding proteins help in keeping the strands apart, as in when the topoisomerase keeps moving and when the strands keep splitting, they help in preventing the strands from rejoining. So you can see there is one strand from the parent strand and the other from the daughter strand. Therefore, it is known as a semi-conservative. So once the replication completes, there are two copies formed, and each of them has one from parent strand and another from the daughter strand. That is, therefore it is known as semi-conservative. And as I have explained, these are the elongation phases where an RNA primer is employed and DNA polymerases comes into action, leading into the replication. And the termination, and when does a DNA replication terminate? There are two instances that the DNA termination takes place. First and foremost, it is during when it reaches the end of chromosome, and the other instance is once it reaches the other replication fork. When two replication forks meet together, it terminates immediately. And RNase H is a very important enzyme which helps in removal of the RNA primers, and DNA ligases joins the discontinuous Okazaki fragments, which I was speaking a while ago. And so the key important concepts that we have learned from this particular process is DNA replication is a semi-conservative process, and DNA replication is a very well mediated process which employs a lot of DNA polymerases, proteins and different enzymes. And to primer starts the primer which is placed by the RNA primase is the start of the replication and leading to the synthesis of DNA from 5' end to 3' end, the daughter DNA. And during the replication, there are two strands which are formed. One is leading strand and the one is lagging strand. And finally, there are different types of enzymes like DNA polymerases. There are different DNA polymerases which we will be seeing in the future class, and there are DNA primases and DNA helicase, ligase and topoisomerase. Likewise, there are a lot of enzymes which are employed in the process of replication. Thank you.